So let's see how this nano goes. Actually, yes, this is a talk that is really a patchwork of ideas that really came, mostly came as a reaction to the discussion that we are having these days. And so instead of the typical introduction that I am used to give, I collected a few pictures and a few provocations that particularly touched my attention. And so, okay, yeah, I will be talking about yielding, and I probably don't need to say anything about that. But with all these questions that we are being asking about this phenomenon, how ductile, brittle, fragile it is, how if it is localized on a macroscopic scale or not, if it depends on the size that we are looking at, if there are correlations, which is also related to the partial correlation, and then underlying there is Ellen's comment or about we measure what we want, right? So all this, all our way of talking about this is probably biased by what we can measure, which is not everything that we would possibly like to measure, right? So the question behind all this, so the unifying thread that I would like to take is what is the relevant length scale that we should look at to describe yielding in a proper way. So macroscopically, the entire system eventually flows, and that's what we call yielding. But what is the scale that we should probe to understand why it happens, how it happens, when it happens? There are a few suggestions, right? Starting from the very local particle scale. Peter Schall has shown that there are these sort of elementary rearrangements that eventually lead to flow sometimes, but it has also been shown that these rearrangements often, if not always, are organized. They are not happening sparsely, or maybe sometimes they do, but they also can be organized spatially in bands or in heterogeneities. There can be correlation depending on the interactions, and this definitely has an impact on the final properties, because this is a CD case under birefringent light, and you see that there are stresses stored in this solid material after it has been fluidized and cast. And having or not these sort of stresses is important, because if you heat it up, it will deform. Cement will eventually crack if you don't heal the stresses that are in it. And there are many, I am learning right now that biological systems depend on their tension that is stored in it otherwise. But what is the length scale where these stresses are stored? Is it the cage, very localized cage of neighbors? Is there a mesoscopic scale that we should look at? And it is very difficult to probe.
Sorry, can I just ask?
Yes.
Using the top photoelastic image for anything? Or it's just a picture?
I am using it for something.
For what?
It's a sort of a provocative...
No, but there's no length scale in that. I just want to tell you that right now. These are power law correlations.
Sure.
So there are no length scales in them.
Gramblers.
Correct. So if you're showing it, then, I know in those systems, there are no length scales.
Yeah.
And you can't melt them or add temperature to them.
I meant to be provocative.
No, it's not a good way to provoke because it's not rigorous. It's not a good way to provoke because it's not rigorous.
I understand.
Yeah. So I think people should stop that because it has nothing to do with any length scales.
But if I can iterate on this, it's the usual way I have been taught to think about soft glasses really come from thermodynamics and that is really cage-based. Under some conditions, what I see is that there are correlations of local forces that start to look more like this experimentally.
Correlations that decay as power law.
Sorry, can you please?
That decay as power law. Stress correlations decay as a power. There's no length scale in it.
In all materials?
In all elastic materials.
Yeah, but a soft glass is a viscoelastic material, so it has dissipation. Yeah, I know. I don't have anything like that in a viscoelastic material, so I just want to poke on the fact that these mesoscales are neglected, but I'm not entirely sure that is right to neglect. But yeah, I mean, it doesn't need to be rigorous. It wants to be exactly this, is minestrone alla milanese. It's a dish where you put a lot of things inside that basically have nothing to do one with another, but then when you taste it, it's good. I hope this talk will taste good. So I will start from the other end of the spectrum, the one that I'm skeptical about. So, and one is very popular in the lab I come from. I come from a lab where people really compare particle-based simulations with rheology, and they are very happy about that. So they have this sort of simulation box, about 1 million particles at most, and then they have very precise ingredients about interactions and everything. And giving the precise ingredients, they can produce flow curves that look basically great, identical to rheology. And they are very happy about that. They say, oh, okay, we know everything about the system. We know that if we prescribe these interactions at the level of a particle, we explain all we need to explain. And they explain it in this way. I mean, this is one example. This is a pair correlation function between two nearest neighbor particles. And they show that the distribution is skewed when you go through an overshoot, and then it is released. So really looking at a very, very local scale, you have the right rheology. And what the physics is that deformation, yes, applies a strain to the local distribution of neighbors, and plasticity releases that. Two ingredients, all you need to know, but maybe not. So that is the ground truth that I want to start from, and to explore if there is anything else that we can add to our soup. And this anything else, well, following Hélène's comment, is not easy to measure, because we are talking about sub-micron particles that undergo nanoscale displacements, and we are talking about centimeter scale samples. So you have a lot of orders of magnitude to bridge to really see if there is anything going on at some scale, at some possibly relevant scale. So this is what I like to do. This is the instrument that we built at the SPCI. It is a sort of a rheometer with sliding plate. The plates are transparent. There is a fluorescence microscope mounted on top. These can be used to reconstruct flow profiles resolved across the gap, to check whether or not you have banding slip and all these fun events. And coupled to this also, there is a sort of an imaging system, but it's a photon correlation imaging system. So it's a sort of a hybrid version between a microscopy setup and what we have seen the first day as a scattering, speckle scattering instrument. So the typical image you see is like this, is a real space image. So you see bubbles and you resolve them spatially, not on purpose, but there are always bubbles. But on top of this, you see this speckled pattern. It's a sort of a QR code that encodes the position of all the particles that are in the beam. And that you get, and therefore you get the microscopic non-affine dynamics. You have a laser, you collect light from there, and yes, and this light forms, I will not repeat this. And this motion of these speckles tells you about, if you compute the correlation of it, the time correlation of it, you learn something about the relative displacement of every single pair of particles averaged. Yes?
It's quite average.
It's averaged throughout the gap. That's right, and that's important, thanks. It's important, exactly, because there is a trivial direction in which you have a lot of relative displacement, just because one plate is fixed and the other moves. And another direction that is very interesting, because there is nothing that you expect unless there is something interesting. And see, this is because, and the light scattering gives you a mean to decouple these two, because it projects everything on a vector, the scattering vector, that is set by the geometry. So what we do is to use two vectors to reconstruct the two-dimensional motion, using two lasers, and which give us two vectors with the same sensitivity, but along two different axes. So I would like to start from the simplest experiment that you can do in this way. And the simplest experiment is just to shear at a given speed. If you start from a solid-like sample and you measure the stress over time, this goes through an overshoot, and then relaxes to a plateau that is the viscoplastic flow of the material. And this is what your particle-based simulations would tell you. And then we measure correlations. So this is a correlation. This goes from 1 to 0 in a time. This is a time delay between the two states that we are correlating. This time is the time it takes for particles to move off 100 nanometers, more or less, in the velocity direction and in the vorticity direction. You learn something from this, but you even learn more if you compare different strain rates. In this case, you get all these from low blue to high strain rates red. And you try to plot things not against time, but against time rescaled by the rate. So you get good collapse. This shape along the velocity direction is trivial. You get it with an analytical expression that has no fitting parameters. So basically just tells you that there is no shear bending and no slip. It's a good sanity check. And this one, it comes from the integral of all the relative displacement along the gap. So the speed of particles across the gradient is a fine yes. Along the vorticity direction, things are a little bit different. They still scale with the shear rate. I didn't fit anything. I just plotted an exponential function here that is a diffusive dynamics, where the diffusion coefficient is given by the particle diameter and the yield strain. This tells you that every time I reach the yield strain, particle will, locally in the cage, particle will rearrange in a way that has no direction, that has no preferential direction, but makes a jump that is order particle size. So it's not perfect, it's good, and I would, yes?
Dimensionally correct. So is there something else that you...
Yes, this is diffusive in strain space, because this is a strain, so the parameter is not time, but it's a strain. So it's a sort of a pseudo diffusion coefficient. Yes?
What system are you looking at?
Oh, sorry, yes, I keep on forgetting to tell you that. It's a dense suspension of microgels.
microgel?
300 nanometers at a density that is in the jumping regime. Yes, so this slide is to tell you that all I said at the beginning is not entirely wrong. So particle level, theory that are based on the single particle environment do capture a lot of what is going on. In particular, the viscoplastic flow doesn't look like, there is nothing really that I would like to know more. It looks like it's a complete information on this. Things start to be different when we look at what happens that brings to that, so the yielding transition. So funny enough, okay. Let me go a little bit fast on this, sorry. Otherwise I will not get to, what I would, okay, the message of this slide is, you do see, so what for instance, Suzanne would suggest is that there might be shear banding there. And what we saw the first day of the XPCS results is that there is some sort of backflow. And indeed, and we were looking for it 2 days ago. So it's a very recent thing. Provoked by this, we tried to look at it, and this is the affine, a time trace of the affine correlation. If everything was affine throughout the experiment, we would see a straight line. We don't see that. We exactly at the peak, we see some sort of lack of affine deformation, as if indeed something was backflowing or shear banding or slipping or a mixture of those. And scattering, it's hard for scattering to tell anything more profound than this. But luckily we have the PIV. We can look at flow profiles, and we were very excited, and we saw nothing. Really nothing. So this is one. This, I don't know. Probably this will not tell you anything, but I will. These are speeds as a function of position across the gap for different times. It's hard to tell, but if I do a time trace like this of the deviation from between the speed that I measure and the speed I expect for a fine deformation, I plot the deviation of these for different positions in the gap. As a function of time, nothing. So it globally, it looks like it's perfect. I was optimistic. I was looking for it. I cannot tell I found it. So I don't think we can say it's shear banding. I think there is some sort of backflow still, because there is less affine deformation, but that backflow doesn't look like this backflow is really organized in a coherent way across the gap. It's more of a local backflow or a mesoscale backflow. And this is interesting, but it's hard to probe it in these transient experiments. So for this reason, we went to oscillatory experiments. Oscillations are good because you can average on a lot of, so statistics get significantly better. These are viscoelastic moduli as a function of amplitude. We saw that in a bunch of different forms. These are the Lissajous curves, stress, strain. And these all look very much like what Simon showed us. And they really look like Carbopol is another microgel. The picture that I get is that it's a low brittility system. So there is nothing very catastrophic, gentle way of yielding. If I use this model to not fit, but to superimpose to my data, looks okay. Maybe brittility is not 1, it's a little bit more than 1, but in a sense, it's okay. And then what I can learn new, I will tell you this, and then I will skip a couple of slides to show you the exciting part. Well, I don't know what you will find exciting. You will tell me. But what we can do with oscillations is that we can start from a reference time point and then correlate all the other phases of the oscillations to this starting point to start addressing the question of whether this dynamics is recoverable or not. We see dynamics. Is it relevant or not? Well, probably the answer depends on whether it's recoverable or not. And this is a way to probe it. And so we do that in the direction of the shear. And this is, once again, not very exciting. We see that the correlation gets lost because I start moving. And then it goes back when I'm again at zero strain. And then it gets lost again. And it is very, very well fitted by the affine theory. Nothing special. On the other direction, it's more fancy, because for a pure affine deformation, I project things along the scattering vector perpendicular to it. I expect nothing. I expect a flat line on 1. I don't see that. I see a loss of correlation, which means that things are moving in that direction as a consequence of strain. So there is non-affine motion, but this non-affine motion is recoverable, is reversible, because correlation goes back to 1 when deformation goes back to 0. So whatever I do in this quarter of cycle, it's perfectly reversible. It's solid-like. It is not an unrecoverable way of accumulating the deformation. Yes?
The normal stresses there as well? Because that's what they would look like.
Normal stresses.
Yeah.
Nice. No, I mean, no, it's not true. We can measure normal stresses, but we need to give up something. So we need to change a little bit the mechanics of the system.
Yeah.
But we can do that. If there is a good reason to do that, we can.
Yeah, okay.
You're looking at the good reason, right?
Okay. Do you have a strain sweep? Can we know where the linear regime is?
Sorry?
Do we know where the
Oh, yeah. That's the next slide. Yeah.
2%.
And okay, so let's go to the next slide. I just show this because this is something that has been predicted on glasses by Jean-Louis Barra and co-workers, just based on the fact that the material is a little bit heterogeneous, at least mechanically heterogeneous. One expects that when you deform it, the stiffer parts deform less, the softer part deforms more, and overall you get something that is non-affine. But then this is just elastic. So when you go back, stiffer parts recover less, softer part recover more. You go to the beginning. But then back to the question: where are we with respect to the strain sweep? And then we are here. So we are in the linear regime. And then if we go here, which one would have? Okay, rheologically, this is not the cleanest data ever because it's a shear cell, but one would also call it linear regime. No higher harmonics. Things change significantly in the sense that these non-affine deformations are not that, or are reversible in a stroboscopic way. So they are reversed after a full cycle, but they are not reversed after half a cycle. And that is what people call reversible plasticity. There is a path that things do that doesn't go back after half a cycle. Half a cycle I do something, the other half a cycle I go back, but from another path. And this is the signature of this. So it's not impacting rheology a lot, I would argue, but it's there, and we are sensitive to it. And reversibility is lost only at a second time. So there is a sort of a spectrum of things that one could possibly look at. And let me skip all of them, apart from the last one. So it's a pity. Okay, let me go a little bit faster. Let me focus on the onset of irreversibility, because this is what yielding is about. How I dissipate of unrecoverability, using a rheology term. How do I probe it? Well, in this plot here, sorry, I am plotting the height of this half a cycle point and the value of this full cycle correlation point. And here you see that the half a cycle point decorrelates first. This is the reversible plasticity. And the full cycle point decorrelates a little after, which is the onset of stroboscopic irreversibility. So there's a lot going on. I'm focusing on the onset of stroboscopic irreversibility now, which is very subtle. It's a tiny decrease with a lot of rheology change. So there is not much that you need to do to change rheology, which means that not all particles are probably important. So you don't need to rearrange everything to have yielding. But this is still not very easy to detect. And so we do many cycles. If you do many cycles at this amplitude, you get a correlation that goes up many times, and you still get, after 50 cycles, you are still totally correlated to your starting point. Then if you increase it just a little bit, you start correlating up almost perfectly, but after a while, you lose correlation. It's a sort of a fatigue, although the term is not very precise. And then even more when you increase, right? This is a transition. So studying the decay of these so-called correlation echoes, because they come back periodically. So studying the decay of these correlation echoes, we came up with a model that I will skip, because but it's published. So this is the only thing I managed to publish recently. So we came up with a model that tells you that this yielding happens by a sort of a combination of two types of dynamics. One is solid-like, one is liquid-like, and these two dynamics coexist at yielding in the region where you have the stress overshoot. And this coexistence tells you something, because this coexistence you don't get if you only look at a single position in the sample. This would be you don't get it. To get this coexistence and this that well-defined transition, you get one second element that we developed in a sort of a lattice model. This second element is correlation between positions in the sample. What the community in the glass community calls dynamic facilitation. And so if you introduce these correlations, these dynamic correlations, then you have a lattice model that can reproduce your data very precisely, and you also have a mean field solution to this that is nice because it looks really like van der Waals equation of state for real gases, where the microscopic rate of rearrangements is the volume, the applied field is strain instead of pressure, and these temperatures have some sort of maps, some sort of notion of glassiness. And so okay. Yeah, let me skip this. No. Let me just say on this that like the van der Waals equation of state, you can describe things in terms of a diagram, of a phase diagram that predicts a liquid phase, which here will be the solid-like behavior, a vapor phase, which will be the fluidized behavior, and a sort of a transition between the two that is yielding here for us. So the two models predict the same phenomenology, and the part I am interested here is the existence of a critical point where typically fluctuations, heterogeneity, and all of these phenomenology are very relevant. And so we really looked for it. All these spatially resolved experiments are meant to look for this sort of heterogeneities and correlations. And so we said, let's just look if we can get huge fluctuations near what we predict as the critical point.
When you're in the unstable region, they separate you in a fluid state and a vapor state, separate by one interface.
Yes.
What does it mean in the yielding? That you separate in a solid state and a liquid state, separate by a boundary, or is it shear band?
That would be sleep or shear band or something. So a shear banding transition is exactly this. You have half and half, so we don't know. This is why we did the experiment and the next slide. But before then, we can solve the model. We can solve the lattice model in simulations, and this is what the simulation suggests. This is a sort of a. I am increasing the strain amplitude here, and I am plotting the spatial distribution of local rates. So they don't segregate they don't always segregate, at least, into macroscopic phases with an interface, but they still segregate locally in distinct regions with relatively sharp interfaces. And this is what we are looking for.
Figure said dynamics.
Oh, yeah. This one. Sorry.
What is it? What does that mean?
Dynamics is what we probe with the relative motion between the rate at which things move relative to each other in the sample. It's a very vague term and depends on what is the sample that you are looking at. In this case, you are really looking at displacement between colloidal particles.
So amplitude. So this is a full period of oscillation.
Yeah.
Are you showing an average
No,
over the period?
I am showing one single period. So the stroboscopic, it's stroboscopic. So I look at how unrecoverable things are after one period.
You're in the unstable state, it's time dependent. So here you show an image, but at what time?
Unstable here?
Yes.
Oh, yeah. There is all a transient to reach what we can call a steady state that can be very long. And this is the steady state. So you oscillate for even hours. You still get something that evolve and rearrange, but at some point you reach something that you can claim it's steady. And this is what it is. Sorry.
So this rate is going down this way, right? I was getting confused.
No. Yeah, sorry. It's true. These simulations, this is a time. So rate would be 1 over that. The rate is increasing. This is 1 over the rate. Sorry. Oh, strain is, yeah, I didn't know. Okay, yes. The strain is going down because it's what the mean field model tells. So its pressure is mapped onto 1 over the strain. So if I want to keep the visual analogy, I need to put the strain downwards. And the strain sweep would be starting from the solid and increasing the strain. So going down the isotherm like this, reaching the phase boundary and then yielding, and then shear thinning like this. Oh, because like van der Waals predicts state of the matter that never melt or never evaporate because they are not liquid to start with. In this case, you have state of your soft glassy, state of your sample that never yield because they are never solid to start with. So these describe all the liquid systems that are liquid even at rest. I will show you just the last thing, and I will skip all this. well, yes, this is the thing I want to show, but I will show you big. So this is last thing promised. This is a spatial map of local dynamics, stroboscopic, in a glass. So here you see bubbles. Around bubbles, things are a little bit faster. This is the scale. Correlation 1 means after one cycle, everything has been correlated. 0, everything has been uncorrelated. And this is a map, right? So bubbles nucleate little unrecoverable regions around, but they are very localized. And up here, there is a triangle where I look at the affine deformation. So these you don't need to look at. Other than that, this is all white, no dynamic heterogeneities. 8%, 12%, 16%, some granularity, 21, 26. So you start seeing, and you are climbing down the strain sweep, right? So now you are in the van der Waals isotherm, you are going down and approaching the phase boundary, the coexistence region. And so you are nucleate, and so at some point you do that. So at some point you see that things become significantly more, so up to here, it was questionable whether or not these were real heterogeneities. Here it's clear. So you nucleate heterogeneous fast rearranging regions inside the material. And this is 1 millimeter. So we are probing tens of nanometers displacement. And those are heterogeneous over millimeters. That is why it's not easy to probe them. And then those evolve into mostly rearranged regions until you are left with everything that has been apart from the bubbles. And yes, let me stop here. And thank people from my previous lab and my current lab and you for your attention.